Hello, my name is Yosuke Tanigawa, and I'm a PhD student working with Manuel. Here we want to present our Decomposition App for interactive exploration of latent structure of genetic effects across disease outcomes, biomarkers, and lifestyle measurements. If you go to this Decomposition App page on our global Biobank engine, you will have an illustrative summary of our approach and sets of results using different conditions. The main challenge in extensive phenotyping combined with the genotype measurements from biobanks is how can we understand and infer the relationship between multiple phenotypes and multiple variants. To answer this question, we analyzed more than 300,000 individuals in UK Biobank cohort and characterized genome-wide association studies for more than 2,000 phenotypes, including disease outcomes, biomarkers, and lifestyles. We took Z-scores from genome-wide association studies and constructed a summary statistics matrix. We applied truncated singular value decomposition for this matrix and obtained three matrices, U, S, and V transpose, which corresponds to phenotype singular vector matrix, eigenvalue matrix, and variant singular vector matrix. Using these three matrices, we can compute two types of scores. We call as squared cosine score and contribution score. By looking at this square cosine score, we can identify which components are the key for a given phenotype or a given variant. Once you identify the key components, you can look at the contribution score and ask which phenotypes, variants, or genes are the driving factor for that component. And you can come back to the square cosine scores for the other phenotypes or variants. The Decomposition App, which we present here, provides an interactive exploration of these scores to understand the latent structure in genetic effects. We'd like to show you an example using one data set, here, this one. If you click this, we will navigate you to the Decomposition App. For this particular data set, we are looking at coding variants outside of major histocompatibility complex or MHC complex region. Okay, now you are on the Decomposition App page. Here we have squared cosine score and contribution score, as we saw in the graphic overview of the approach, and several other plots. Let me explain these plots one by one from top. Let's say you are interested in asthma. You can search for a phenotype of interest using this search bar. Asthma, click this. Then you will have a phenotype square cosine score for asthma. So this plot basically says PC59, PC35, PC52, and PC56 are the key components for asthma. Let's click on PC59 and go down a little bit in this page. So now you selected PC59, and this is the component you're interested in. For this component, we have phenotype cosine score and variant contribution score here. If you look at the phenotype contribution score, you see eosinophil percentage, asthma, eosinophil count as a kind of top 3 driving factor for this specific component. You can also look at variant contribution score and find that, this variant on chromosome 15, the variant on chromosome 4, chromosome 6 are the driver for this component in variant space. If you click at one of the dots in this plot, let's say this one, you will have a squared cosine score for this specific variant. So you can easily go back and forth between squared cosine score and contribution score. One important characteristic you should remember is having a high score in contribution score does not necessarily mean that the same PC pops up in the squared cosine score. For example, you will see eosinophil percentage as the top hit in PC59. Let's click this phenotype and go to the squared cosine score for this phenotype. You will see the other components like PC12, PC10, PC11 are the key components for this specific phenotype. PC59, which we are looking at, is, I guess, fifth one for this specific phenotype. So the measurements in squared cosine score and the contribution score is one-sided. It's not the kind of bidirectional mapping. So that's why you need to look at both squared cosine score and contribution score. The interpretation of phenotype contribution score is relatively easy because you can understand which phenotype is it by looking at the level of the dots. However, the interpretation of the variant contribution score can be tricky because I can't personally remember everything for name of the variants, name of the genes. So we aggregated a result of the variant contribution score by genes and created another plot here. So basically, this is contribution score, but we aggregated by gene. So you can see that this gene, USP8, ADH1B, CALMIL1, KIA1109, IL33 are the driving genes for these specific principal components, 59. We actually, IL33 has a protective hits in GWAS studies. So clearly this is capturing some genetic structure, latent structure in genetic effects in our phenotype of interest. We also have similar plots for phenotypes, which we see eosinophil percentage, ADMAR, eosinophil count, and so on. If you click on this radio button, like open the phenotype page on click, or open the variant or gene page on click, you can easily navigate to our phenotype page. Like if you click on ADMAR here, you will have a phenotype page loaded for ADMA. For if you click on the specific gene, like IL33 here, you will have a gene page loaded from this link. It will take a while because the internet connection in my laptop is currently a little bit slow. But in this way, you can look at the this decomposition analysis to look at the relationship among different phenotypes and variant. But at the same time, you can also look at the marginal effects of variant to a specific phenotype, like one phenotype versus multiple variant analysis, like Manhattan plot and list of variants we use in the study with different filters or different sorting columns. So this is nice. And same thing for our gene page, like we have some summary of our findings, tables and plots. Okay, let's go back to our decomposition page. Another way to interpret this singular value decomposition is basically you can look things as a PCA. Sorry, before going this, I need to explain these plots. So you may notice that this specific principal component has multiple phenotypes and multiple genes as a driving factor for this specific components. You may wonder how many phenotypes or how many genes are contributing to these components. To address these kind of questions, we prepared global summary of contribution scores, which we're presenting here. Each color in this plot corresponds to one particular phenotype in our study. And we only showed phenotypes with more than 1% of contribution scores and aggregated the rest of the phenotypes as a letter on the top here. So based on this plot, you can say that for each component, for most of the components in the top 100 components we identified a study, there are infinitesimal effects from multiple phenotypes, except with the exception like these components and these components. The same thing here for same things for genes is presented here. Like you can see, small effects from large number of genes for almost all principal components we identify in our approach. You may notice that I'm using principal components to refer to one component identified by SVD. So this is true because one can look at this SVD as a variant of PCA. Well, the two-sided version of PCA. So you can plot the two principal components, let's say PC1 and PC2 on the phenotype space on the left or variant space on the right. So if you look at these outliers in PC2, you may find related phenotypes. For example, this one is reticulocyte percentage. This one is reticulocyte percentage also. Reticulocytes count, reticulocytes count, and reticulocyte fraction. So our approach is clearly capturing some relationship among phenotypes and some relationship on variants. Like for another example would be this is standing height, sitting height. So these are actually the kind of key drivers for PC1, as you see in this one. Well, I should say this one. Sorry, PC1. I should click it. Yeah, this one's standing height and sitting height. Let's go back to PC plot. Of course, you can look at scatter plots for different pairs of PC. You can click by scroll button and make a new plot. You can save the figure if we want by clicking on this camera button, like to download the plot as a PNG format like this. And the last plot we are presenting here is script plot or variance explained plot. This is basically answering the question that how much variance in the original matrix is explained by this specific component. For example, PC1 explains almost 6% of the variance, PC2 explains almost 4.4% of the variance, and so on and so on. So in summary, we believe that our decomposition approach is a first attempt to understand the latent structure of genetic effects across multiple phenotypes and multiple variants. And we propose that two scores, like squared cosine score and contribution scores, are useful to investigate that structure from the data matrix. And we have the decomposition app, as we're showing here, to have an interactive exploration of the data we have pre-computed for you. So feel free to explore, and we believe that this will accelerate some therapeutic hypothesis generation. And we are looking forward to hear interesting story from you. Thank you very much for watching the video.